Hello everyone, and welcome to today's webinar, Role of SARS-CoV-2 Serology in a Vaccinated World. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thorson. To learn more, visit thorson.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, David Engman, MD-PhD, Medallion Chairman Emeritus of Pathology, Cedars-Sinai Medical Center, Visiting Professor of Pathology, UCLA, and retired Professor of Pathology at Northwestern University. You may now begin your presentation.
Everyone for joining today. I'd like to discuss the role of SARS-CoV-2 serology in a vaccinated world, a very important issue as we move through the end of the pandemic, hopefully, and on to a post-vaccinated world. The objectives of my presentation are as follows. I'll first give a brief update on SARS-CoV-2 and the disease COVID-19, review the current and emerging applications for serology to this virus, discuss the variation among the different vaccine types with respect to 3 different major issues, and finally talk about a new innovation in serology which involves the natural form of the major surface antigen spike protein as a capture antigen for serologic diagnosis. First, an update. So remember, just very briefly, one viral particle can enter a host cell and through taking over the host machinery and providing some proteins of its own can generate thousands of copies of the virus from a single infection cycle. There are multiple possible outcomes of infection that's acquired through the inhalation of droplets containing small amounts of virus. About 40% of infected people have no symptoms at all. They're asymptomatic. 40 to 50% have mild symptoms that do not require hospitalization. Around 12% of infected people globally require hospitalization because they're very ill with this infection, and 3% die of this infection worldwide. And then a very interesting and unusual and not very well understood sequela of this infection is called long COVID, where 1 or more symptoms of infection persist beyond 6 months after clearing the virus. So this is basically the 4 different outcomes in a very simplified way. SARS-CoV-2 is a perfect virus. It's perfect because it's easily transmitted. It can be transmitted before symptoms develop, if they develop at all. And it only causes serious illness or death in a low percentage of infected people. This is what makes it so successful. In contrast, its cousin SARS-CoV-1, which was circulating around 15 to 20 years ago, was not perfect. It was too lethal. It killed people before they were able to transmit to others, and transmission did not occur before symptoms developed. So people, once they developed symptoms, could sequester and then prevent spread to others. So this is the difference between SARS-CoV-2 and 1. At this point in time, after this pandemic began in late 2019 and all the way to the present time, we've had waves of virus that have hit different parts of the world, shown in different colors, with the Americas in yellow having its surge. Southeast Asia, especially India, is having its surge now. Europe in green surged some time ago. And at the very moment, something between 70 and 80,000 deaths per week are still happening with this pandemic. So it's still a major public health problem, even as much of the world emerges from this. Epidemiologically, there have been 175 million cases in the world with 3.8 million deaths. And what's interesting here is that that represents 2.2% of the world population, and the mortality is 2.2% of that 175 million. In the United States, 10% of our population has been infected with about a 2% mortality rate. Canada, about 4% with 1.8%, and India, 2.1% of the population is infected. And this number 1.3 is growing as the India cases lead to deaths in a small number of people. There's still 130,000 cases here out of 432,000 new cases in the world. Now, I should emphasize that in terms of deaths, we still are losing 10,000 people per day of this infection. That's enough to fill 50 200 passenger jets per day. It's still a major problem. What about serology in a vaccinated world? So we're skipping ahead beyond the basic biology and epidemiology to the major focus today. First of all, we need to get the world vaccinated. At this point in time, we're approaching 3 billion doses of vaccines that have been administered on Earth, with some countries in the darker color here doing better, United States and Canada, China, Mongolia, and much of Europe, but other areas not doing so well with vaccine delivery. In the United States, 44% are fully vaccinated, 53% have had at least one dose. Canada is doing a little bit better with at least one dose, but 15% because many people haven't received their second shot. And India is just getting started, 15% with one dose, 4% fully vaccinated. And this is part of the reason that we're seeing a big surge of cases and deaths in India at the moment. The comparison of where vaccines have been done very well and the current hotspots reveals some very interesting things. First of all, in South America, there are many cases going on right now, and the vaccinations have lagged a little bit behind what we see in North America, Europe, and China. Whereas Africa, the African subcontinent, is relatively unvaccinated, and it doesn't have lots of cases yet, but there's a great fear that these cases are going to really skyrocket before the vaccination can be delivered effectively. And that's something that the public health community is very concerned about. How about the serology applications? I think about this, and others do as well, as 5 different buckets, if you will. And we'll deal with these in turn, one at a time. The first is primary diagnosis of infection. Now, most people are diagnosed officially by RT-PCR, or detection of viral RNA in an upper airway sample. And serology, though, is going to be essential after viral clearance, because some people will be infected and recovered. They may have asymptomatic infection, not even be aware of being infected. And for those people, serology will be the way to determine whether they have had a primary infection at some point in the past, because virus is no longer present after they've cleared it. As you can see, the RNA and antigen peak after about 7 days and then go away sometime between 2 and 4 weeks after infection for most people. But the antibody response, IgM in blue and IgG, arise and persist, and that IgG antibody, which persists, is what can be used to detect previous infection. Epidemiologic studies are largely done through reporting by various health departments based on those RT-PCR results to detect viral RNA. but serology will be more important over time as this pandemic continues and eventually resolves. Measuring immunity in immunocompromised individuals who might not make a very strong measurable immune response to infection or to vaccination. Many studies show that antibody and T cell responses are blunted, and I'll talk about this more later. And we'll discuss this below, especially after we talk about immunity to vaccination. Evaluating plasma for convalescent plasma therapy. This is where you administer plasma from someone who's recovered or recovering from infection in hope that delivering those antibodies to patients who have early infection might help them recover more quickly. The problem is that these convalescent plasma transfers have not worked very well, and monoclonal antibody therapies have not worked as well as hoped for either. As we'll discuss below, the titers of antibodies in vaccinated people are much higher than is seen in those who've recovered from infection. And this gives rise to some hope that there's plasma from vaccinated people may work better than convalescent plasma. Assessing immunity after infection or vaccination. This is one of the major uses of serology going forward. How long does immunity last? We're getting some clues about this from people who were infected a year ago and have been followed through vaccine studies or other kinds of clinical studies. It's not known exactly. How can we measure effective immunity? Are there measures of immunity that would predict whether someone is resistant to reinfection or at least resistant to getting ill requiring hospitalization? Are there specific markers of effective immunity? Antibodies to which antigens of the virus, or epitopes, so which are small parts of those antigens? Is it to the spike protein, which is on the surface, or nucleocapsid protein, a very abundant protein that surrounds the nucleic acid inside the viral particle? T cells, these are lymphocytes, and this is how immunity to tuberculosis is measured, because the antibody response to TB is not a very good measurement, a reliable measurement of immunity. Are there other markers in their blood, or some combination of these various markers? And finally, how long will immunity need to be assessed over time? Is it going to be forever? Maybe until this pandemic resolves and the virus isn't circulating anymore. These are major questions that we're asking now. And I would just mention that there's a lot of work on identifying markers in the blood that might indicate effective immunization. These are beyond antibodies, other kinds of things that the body produces that might indicate effective immunity. And also, people are studying the repertoire of antibodies and T cells that are in the blood that might indicate effective immunization. Not just the quantities of antibodies, but the actual types of antibodies that are produced. So there's a lot of ongoing research to try to get a handle on what are these indicators of successful immunity and resistance to reinfection. It's possible that vaccination-induced immunity will be long-lasting. And some evidence for this is that first SARS infection, CoV-1, which happened, again, 20-some years ago, nearly 20 years ago, they do wane over time, but the T cell responses remain after 15 years, which might allow someone to mount a very quick restoration of immunity in case they were re-exposed to that virus. Problem is, of course, CoV-1 is not circulating anymore, so there's no way to know for sure. The same may be true of COVID-19, and there's indication that infected people have in their bone marrow, which is where cells of the blood are formed, plasma cells that are SARS-CoV-2 specific that last a long time and might provide this long-lasting protection. A real problem happens, though, in cancer patients. And so what happens in hematologic malignancies, which are leukemia and lymphoma, these are cancers of the blood, that only about half of people in this study of 67 patients with hematologic malignancies who received either the Pfizer or Moderna messenger RNA vaccines, only half produced significant antibodies, half didn't, 46%, which might mean that they're not protected in case they're exposed to the virus. And there are many studies in many centers that indicate through their own preliminary results that vaccines may not protect patients with weakened immune systems from cancer, inflammatory bowel disease, or other kinds of immunocompromising illnesses. In addition, a recent study that was the subject of a LabRoots presentation by Dr. Hans last week indicate that while normal controls mount very nice antibody responses to vaccination, that patients who've received kidney transplants have very poor antibody responses upon vaccination at 14, 28, 36, and 58 days post-vaccination. And even hemodialysis patients, patients who need to have their blood cleaned periodically to remove toxins because they have decreased kidney function, they also do not have the kind of strong antibody response seen in normal individuals. And the reasons for this are that kidney transplant patients are on immunosuppressive medications that keep their immune system from rejecting their transplanted kidney. And hemodialysis patients, part of the reason you have dialysis is to remove uric acid, and they have uremia in the blood, which actually inhibits the quality of the immune response. There are many other conditions, autoimmune diseases, immune deficient states, and immunosuppressive medications that many, many people take for many reasons. And these also impair adaptive immunity and might prevent someone from becoming effectively immune after vaccination, and therefore may be susceptible to this infection. So those are the 5 major areas: primary diagnosis, epidemiology, immunity in immunocompromised people, evaluating plasma, and assessing adaptive immunity after infection or vaccination, that are the focus for a lot of the current and future serology. I'd like to now turn to discussing the variation among vaccines with respect to prevention of infection, adaptive immunity, and effectiveness against the variants. First, prevention of infection or serious illness. All 5 major vaccine types work well. Messenger RNA vaccines by Pfizer and Moderna, protein subunit vaccine, Novavax, as an example, and viral vector vaccines by Johnson & Johnson and AstraZeneca. There are differences. The percentage protection might differ here and there, but overall, these all work very well at preventing certainly serious illness and death, and also primary infection. So they all prevent infection and serious illness in most people. There are a few percent, 5, 10, 15, 20%, depending on the vaccine, that still can have breakthrough infections, but by and large, they work well. They work well because they induce strong adaptive immunity, and I'll talk about this in a minute, what that means, to SARS-CoV-2 virus. Other vaccines are likely to work well too. How well remains to be seen. But overall, it's been a huge success. One of the greatest successes of modern medicine actually is the rapidity and success of developing vaccines against the COVID-19 virus. How about adaptive immunity? Well, let's discuss what this means first. This is the specific immune response that a human body makes against a pathogen, a virus, bacterium, any kind of infectious agent, a vaccine, or a foreign body, right? It involves both an antibody response and a cell-mediated response. These are both kinds of immunity, 2 different arms of the immune system, and they are both part of adaptive immunity. Lymphocytes are the mediators. Lymphocytes produce antibodies, and some lymphocytes produce to this virus neutralizing antibodies that basically block the virus from infecting cells and sort of stop it in its tracks. Neutralizing antibodies are most often directed against the spike protein, the S antigen, and can be measured by either plaque reduction or microneutralization assays, which are basically in vitro tests that measure the virus's ability to infect cells. And if you add serum or plasma containing neutralizing antibodies, it reduces the virus's ability to infect those cells. And this can be measured in these assays. Unfortunately, neutralizing antibodies alone do not seem to provide full protection from this virus. And the evidence for this is that, as I mentioned earlier, convalescent plasma and monoclonal antibodies, the therapies have been disappointing. Cytotoxic T cells, this is part of that cell-mediated immunity, they can attack and kill virus-infected cells. They have T cell receptors that are specific for viral proteins that are then presented on the surface of an infected cell. These cells recognize those, attack and kill those cells, and they can be specific for many viral proteins, not just that spike protein, but any protein, viral protein produced in those infected cells, and there are many. Just to review a few of these proteins, this is the SARS-CoV genome, CoV-1 or 2, it's basically the same genome, and they produce a number of proteins. This one's called ORF1A, spike, membrane protein, nucleocapsid protein, and E antigen, and these are the four major proteins, S, E, M, and N, found in the viral particle. However, in an infected cell, the virus also produces all of these other proteins that are encoded by all these other genes, and all of them actually potentially induce an adaptive immune response. So adaptive immunity can develop against any and all viral antigens, external and internal to the virus, and even to proteins not found in the mature viral particle that are potentially ways that we can induce these cytotoxic T cells to kill viral infected cells. The S, spike protein, is most often used for vaccination. It's used in all of those vaccines I mentioned on the slide a few slides ago. S and N are most often used to measure antibodies to the virus. This is just because manufacturers just chose these two major antigens over a year ago to develop their initial serologic tests. N is used to detect infection in people who've been vaccinated with spike protein. The reason is such a person will make antibodies to spike that will not allow spike itself to distinguish between a vaccinated and a vaccinated infected person. Both types of people make antibodies to spike, but the N antigen can be used in those patients to distinguish vaccination from infection. There are some differences in the immunity induced by the different vaccines. It's not really known whether those differences matter yet. Again, this is all new territory. This infection's only been around for a year and a half, and we're still learning a lot about it as every day passes. The vaccines do appear to provide effective immunity against the variants. This is likely because each vaccine induces a very strong immune response against many proteins, many viral proteins, so it's polyspecific, and many antibodies and T cells reactive against each viral protein, so polyclonal. This is the way we make immunity to any infection. And so this is, in this case, why the vaccines appear to provide effective immunity against the variants, because the variants might differ in their protein sequences from time to time, but the vast majority of antigens and epitopes, small parts of those antigens, are going to be the same across all variants. And so this is why immunity to the spike protein works very well, even across variants. And it's important to understand that both antibodies and T cells are important in our fight against many infections, including this one. What about effectiveness against the variants? Well, one of the hallmark features of viruses is that they mutate. They mutate because the enzymes that replicate the viral nucleic acid, the viral genome, make mistakes. And those mutations then occur at random throughout the entire viral genome. Some of those mutations kill the virus. They inactivate a protein that's essential for the virus, and those mutants don't survive, and they're eliminated from the population. We never see them in population studies. But some give the virus an advantage in infectivity. They can either help a virus invade cells, divide faster, survive in respiratory droplets. There are lots of ways that changes in protein sequence can help a virus to be a better spreader. And that's what it's all about. When we talk about viral variants increasing and decreasing in frequency, it's because they have an advantage in spreading. That's the only thing that matters, not whether they kill people, but whether they can spread better. Some of the mutations are in the spike protein. That's this viral surface protein that binds to the host receptor that initiates the invasion process. That's called ACE2 is the name of it. And scientists have been sequencing viral isolates from around the world. There have been hundreds of thousands of sequences determined over time, beginning way back in Wuhan, China in late 2019. And over the months, you can see the acquisition of new variant sequences through following all these little lines here are different viral types having different mutations. So this is after April of last year, June, August, October, December, February, April, up to June of today. So there's been a huge number of new variants, including some that have not varied too much since the original Wuhan strain back at the end of 2019. So the number of variant sites per virus isolate are small, but they're measurable. They're measurable because we can sequence the viral genomes and then follow them epidemiologically over time. So these variants of concern, or variants as people call them just generically, spread through populations, and they have molecular features that facilitate their spread. At the moment, there are 5 that have been labeled by the WHO, alpha, beta, gamma, delta, epsilon, that previously refer to variant names as followed, where their initial expansion occurred in the United Kingdom, South Africa, Brazil, India, and California. And by tracking these over time, you can see, at least at the moment, that the B117, that's that alpha, the one that originally initiated in the United Kingdom, is the dominant form with about 70%. You can see that even better over here. 70% of all the current isolates are of that type. But I want to point out that the Delta variant, this is the one from India, is currently 2.7%. This little tiny dark red one here. And that was not even 0.1% even a few months ago. So over time, these variants can increase or decrease in their prevalence, in this case in the United States. So some spike proteins mutations allow the virus to enter cells more efficiently. We're talking about how they work. You know, what is it about the variants that make them expand? And some variants contain other proteins that are produced in the infected cell, in this case ORF6 and ORF9b, that interfere with host immunity and are expressed at higher levels in some variants. So it's a combination of specific protein mutations that allow the virus to do its thing better, and viral proteins that interfere with our ability to fight the infection. That seems to be how they can work. How well do the vaccines protect against the variants? Very well. There have been many papers, and this is just one, for example, that shows that the Pfizer vaccine protects against the 617, which is the Delta from India, that initially arose in India, and the 351 variant, which is the one from South Africa. So The Pfizer and Moderna vaccines were generated against the sequence originally found in Wuhan, China, but they seem to work very well against these variants as well. And the same thing is true of other vaccines. There are, however, occasional breakthrough infections, and that's 5, 10, 15% of people can still acquire infection. And in some cases, they can acquire infection even though they've had very high levels of antibodies. They get very high virus upon infection. So the antibodies were present before infection. Virus gets to the very high levels, high viremia it's called, during infection, and they can get very sick and even require hospitalization. Most people with breakthrough infections are either asymptomatic or have mild symptoms though. So, robust adaptive humoral and cellular immunity develops after vaccination. That's why it works. And it is possible that using 2 different vaccine types might have some advantage. And there's a lot of research going on now where, for example, initially vaccinating with a Pfizer vaccine, but then boosting with an AstraZeneca might give even stronger immunity than just vaccinating and boosting with the single vaccine type. It's also possible that we might induce long-term immunity from vaccination or for people who've been infected and recover for infection. And there's a lot of research on this issue because it's a major question of whether we're going to need booster shots or not. And that's something that we'll learn more about over the next 6 months. Of course, it's possible, and we hope, that over the next months and years that this virus will not be in circulation, and maybe we won't even need to be following this. But for right now, since that's a big unknown, we need to basically operate with the idea that we will need to be surveilling for this virus for quite a long time to come. The next section is to understand the latest innovations in serology assay design, and that is to try to produce a natural form of that spike protein, which is trimeric form, as a capture antigen. So this is where the spike receptor binding domain binds to the host ACE2 receptor. So this is on the host cell, this is on the virus. This binding interaction is what we're trying to target. Neutralizing antibodies can block it. That's the basis for antibody therapy and the plasma therapy. And you can theoretically try to block it from this side or from this side. So people are doing research to try to use ACE2 receptor blockers to inhibit the viral entry. The spike protein itself is a long protein with lots of parts. The receptor binding domain, shown here, is what a lot of people focus on for the development of neutralizing antibodies. But this N-terminal domain is also very important. So these 2 domains are important here. Antibodies are produced during natural infection or to vaccination to both RBD and NTD and to other parts of the viral spike protein as well. The viral spike protein, shown in this electron micrograph, is actually a trimer. It's 3 identical types of proteins, shown here in falsely colored blue, green, and red, that come together to make a trimer. They're sort of all folded onto each other to make a 3-part protein. This is the natural form of this viral protein. So the spike protemer, 1 copy of this, was produced either in its form with the so-called S1 and S2 domains comprising a serology test produced by Diasorin called S1-S2. The receptor binding domain, which is a key part for that binding to the ACE2 receptor, is the protein that's produced by a number of other corporations. And the new one produced by Diasorin at the moment, there may be other companies producing this in the future, has been engineered to hold together as a trimer, the natural form of the protein shown here. And the advantage of that may be that antibodies to the receptor binding domain bind to all 3 of these types of antigen. the N-terminal domain, which is also found here and found in the Diasorin S1, S2, and the trimeric form, can also initiate antibodies that can bind to them. But there are other antibodies that bind to the interface between protomers, the three protomers, the interface between the blue and the green, the red and the green, etc. And those also can be used for serology. There's a recent study by Benelli that compared that Diasorin trimeric S with the RBD tests by Siemens and Beckman. And basically what they found is that in the case of Siemens up on top and Beckman down below, that in most cases, the test results are the same. Positive in both Siemens and Diasorin, negative in both Siemens and Diasorin. Positive in both Beckman and Diasorin, negative in both Beckman and Diasorin. However, so these are concordant results. However, there are some discordant results. These are results like these 62, which are positive by the Diasorin trimeric protein, that are negative by the Siemens RBD test, this one. And likewise, 6 that are negative by Diasorin that are positive by Siemens. And the same thing is true down here with the Beckman. And so what Benelli did is he basically analyzed those discordant samples by that microneutralization assay, which directly detects the ability of the sample to interfere with viral entry to a host cell. And what you can see is if you take the 6 samples that were positive on the Centaur and the 62 that were negative on the Centaur and look at the neutralization titers, and conversely, take those same samples that were positive by Diasorin, the 62, and the 6 that were negative, you'll see that 2 out of the 6 that were positive both by RBD and neutralization, it's only 2 out of 6, whereas 60 out of 62 were positive by both the trimeric S and the microneutralization. So that indicates, and I'll discuss this shortly, that maybe the trimeric has a little better, greater sensitivity than the RBD alone. And that's something that time will tell, but it seems from this study to be very clear. The other question relates to what happens after vaccination. So this anti-S by the trimeric S antigen can be easily used to detect infection. And in fact, at the time of the first dose of vaccine, so before the vaccine's been given, patients are negative. After dose 1 plus 21 days, 3 weeks after the first dose, essentially everyone's positive and has measurable titers using the WHO, the new standard called binding antibody units. And then after 3 weeks after the second dose, the titers are way up in the 4,000 range. So these are uninfected people. 15 people who had been previously infected were also included in the study. At the time of dose 1, before any vaccine, they were all positive because they'd been infected. And what's interesting is that their viral loads are 131 BAU per mil. But after dose 1 and 2, you can see that it's 12,000, 18,000, and then it drops down here 120 days later. But basically the titers in these 15 people who were infected recovered are much higher than is ever observed in the patients who've never been infected before. So it seems that this trimeric S has a very large dynamic range for infection, for detecting antibodies. So these are my preliminary thoughts about this trimeric S. It has a very large dynamic range. It appears to be more sensitive than the other RBD assays, as evidenced by those discordant results that are positive by Diasorin trimeric S, but negative by Siemens, almost are all positive by that microneutralization assay. And that greater dynamic range and sensitivity may be important for assessing immunity over time. So let me conclude. SARS-CoV-2 is a perfect virus. The vaccines work as well as could possibly be imagined. There is a race now between vaccination and spread, with a handful of variants of concern leading the spread. We're especially concerned about sub-Saharan Africa and India as well. It's not known how long immunity from either infection and recovery or vaccination will protect a person from re-exposure. And the recent results suggest that it may be long-lasting, but nobody knows for sure. And protection also appears to extend to variants. There is no gold standard for measuring effective immunity, but people are using serology as the easiest and most accurate, sensitive, and specific way at the moment, and viral neutralization, which is terrific, but it's not really practical to apply to large numbers of samples. There may be other markers indicating resistance. Will we need, these are unanswered questions, will we need a booster? Nobody knows for sure. Why do some people have breakthrough infections, despite having high levels of antibodies, that 5%, 10%, depending on the vaccine? Will all people need serologic follow-up after vaccination, or are there special populations, immunocompromised people, healthcare workers? Will SARS-CoV-2 stop circulating at some point? So this is all moot. We're not going to worry about this 5 years, 10 years from now, maybe sooner. And it is very likely that serology will be the best and easiest way to assess immunity for many of the purposes discussed today. And finally, it's important that the serologic test be sensitive enough to detect waning IgG levels over time, and the trimeric S protein may provide this advantage over current tests. Thank you all for your attention, and we are hopeful that this pandemic will come to an end soon, and we can all return throughout the globe to our normal lives.
Thank you, Dr. Engman, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Okay, our first question is, where does serology testing fit in a post-vaccine world?
Well, thank you all for your attention, and thanks for that first question. That is the big question. I think that while many people look for all kinds of biomarkers for effective immunization or effective immunity due to infection and recovery, serology is very likely to be what people will be looking toward long-term for measuring the presence of antibodies that can protect against infection. And also those antibodies indicate the presence of underlying T-cell responses that are also important for killing infected cells. There's a lot of history of that kind of approach from other kinds of vaccines and infections that we face. Measles, mumps, rubella, hepatitis, where we receive vaccines, and at least in the healthcare community, the titers of those antibodies are checked over time to be sure that those childhood immunizations still have produced circulating antibodies that will prevent someone from getting infected by measles virus, for example. So I think that over time, serology will show itself to be a very durable way of measuring effective immunity to infection. But again, only time will tell, and especially when people, a small number of people, as long as the virus persists and is circulating, become infected, then studies of those people will be important to determine whether the presence of antibodies detected by serology correlates with either protection or lack of protection. And so that's what I see as the future of serology.
Thank you, Dr. Engman. Our next question. We get lots of questions from staff concerning why they should get the vaccination when they are testing positive on antibody tests from previous infection. Can you please explain the overall benefits and why?
Well, I think that as time goes on, people are realizing that the titers of antibodies, and perhaps even, there aren't as many studies being done on the cellular immune responses, but they're certainly much higher after vaccinating someone who, even though they've recovered from infection and have measurable anti-COVID-19 viral antibodies. So the levels of immunity induced by vaccination are much higher than those induced by infection recovery. Formally, nobody's proven that having lower levels or higher levels of antibodies provide different levels of protection. So it really isn't known. And I think that is why the recommendation has been since the onset that even though someone has been infected, that they go ahead and receive the vaccine, because it is not known yet whether the levels of immunity induced by infection and recovery are protective as well as immunity induced by the virus, by vaccination. I should mention, though, that the immunity induced by infection and recovery is against many proteins of the virus, whereas the immunity induced by vaccination is against only the protein used in the vaccine. I actually misspoke a little bit halfway through the presentation about the kinds of immunity generated. If you are vaccinated with a spike vaccine, you're only going to make immunity to spike. There's no nucleocapsid or any other protein in the vaccine, so you will not be immune to those proteins. So I think that only time will tell whether the lower levels of measurable antibodies from infection recovery are adequate in protecting someone, and over what period of time, people don't know either.
Thank you, Dr. Engman. Our next question is, let me find it. Do you see a medical value to bridge B-cell response, IgG or total Ig, and T-cell response to have a more accurate interpretation and to develop the algorithm accordingly?
I certainly believe that from a research side, that it's very important to do those studies, because I think that the quality rather than beyond the quantity of antibodies, whether they're IgG on, there are some people who have IgM levels, that initial antibody that comes up, that persist longer than in other people. And it's not known whether that reflects any aspect of immunity that might be beneficial. And certainly looking at the cellular responses in a research way is important, because that's how we understand what are the parameters for effective immunity. On a practical level, though, to apply that to a large number of people, there are certainly T-cell assays that measure cellular immunity that are used every day to measure immunity to Mycobacterium tuberculosis, the so-called quantiferon assay that is applied to every healthcare worker, and certainly in the Western world as a measure of immunity or exposure to TB that could be adapted for COVID-2. So I think that on a research level, all of that will be going on. It's going on right now. But to apply that to a larger population is a big unknown question. And of course, the biggest question of all is, if vaccination is really rolled out to the world, like we have the ability to do it, we can make these vaccines, we can deliver them, perhaps we'll be able to reduce this virus to the point that it's no longer a threat, and we won't need to be concerned about that. But that's just conjecture on my part. It's wishful thinking, of course.
Thank you again, Dr. Engman. Our next question, is there a mortality difference associated with mutant subtypes?
There are differences in mortality, but they're not very large. It's not like the Delta variant has a 50% mortality rate. When people talk about a 3% mortality versus a 4% mortality, well, that's a 30% increase, 4% versus 3%. But it's still lowish. So I think that there may be measurable differences. The studies generally, because the number of patients is still fairly small and people aren't doing extensive studies on every patient who does succumb to this infection, it's really not known what the significance for large populations is of the small but measurable differences in mortality induced by one variant or another. Remember, the virus doesn't care whether it kills people or not. And in fact, it doesn't care at all. It's just going along its happy way, spreading faster if it can replicate and persist and all those other things that make spread easier for that variant than for other variants. That's how it increases in its prevalence in a population.
Thank you, Dr. Engman. Next question here. Some literatures are citing the prevalence of Delta variant to 10% in the USA and roughly up to 60% in the UK. Do you agree?
Well, I think that you'd have to look at the individual studies and the numbers of people and samples involved in that and to make a decision. In general, the studies that are published in peer-reviewed literature and are reviewed by experts have already been through that kind of scrutiny and statistical analysis, and any differences that are large differences between study outcomes tend to be due to differences in the populations studied and rather than methodology. I will say, though, that for many infectious diseases, there are differences based on the human genetic background, immunogenetic background in some cases, where some populations may be more susceptible to a particular infection and others more naturally resistant to infection. And so there could be some of that going on too. On the host side, host immunogenetic variation and susceptibility and resistance to this virus that could be partly responsible for different outcomes of those kinds of studies. That's a good question.
Thank you, Dr. Engman. Our next question. What are the advantages of the trimeric S antigen as a capture antigen for serology testing?
Remember that this infection has only been with us for a year and a half, and all the major companies, academic labs, rushed to try to find the best antigens to use for serology, for vaccine studies, etc. They tend, for serology, to focus on the S antigen and N, the nucleocapsid and spike proteins, because those turned out in very early studies to induce the highest levels of antibodies. Even though nucleocapsid's not on the surface of the viral particle, it's very abundant in the viral particle and induces very strong antibody and T cell responses. For practical manufacturing reasons and others, every company has its own reason for choosing either RBD or S1, S2, or now trimeric S. It was easy to produce RBD. It's a small fraction of the S antigen. It can be produced very quickly in large amounts, and infected people tend to have antibodies against RBD, so it was an effective way to measure. Where the trimeric has an advantage over both RBD and the predecessor in Diasorin company, the S1, S2, is that it has lots more epitopes, lots more parts of that protein than the small RBD does. So in theory, it can capture more antibodies produced by an infected or vaccinated person, and that's probably responsible for why it's more sensitive, at least in that Benelli paper, to RBD. So I think that's the reason why there's some advantages that it's more sensitive if it's important to measure lowish amounts of antibody, maybe 10, 12 months after vaccination, as antibodies wane over time. The patient sample will still be positive, whereas the RBD might be negative and might lead someone to receive a booster shot unnecessarily. Again, though, I would emphasize that nobody has correlated or attempted to yet
the levels of antibodies, the quantity of anti-S antibodies, and resistance to or susceptibility to future infections. So we're in uncharted territory, but these are theoretical arguments for using an antigen that has greater sensitivity, which the trimeric S appears to from that study by Benelli.
Well, thank you, Dr. Engman. We are out of time for questions today. Do you have any final comments for the audience?
I would only continue saying that it's just been remarkable. I mean, if when I was thinking about this disease, I've been working on this just like many people in the audience for a year and a half. Back in April, May of last year, I never thought it would be continuing beyond last fall, but here we are. And even though lots of the vaccinated world is going on its merry way, we're still losing 10,000, maybe it's down to 8,000 now, people a day. This is not gone. And I think that with vigilance and with continued advances in healthcare diagnostics and treatments, we're hopeful that a year from now, we won't be having the same kind of lab roots seminar necessary anymore. Thank you.
Well, thank you again, Dr. David Engman, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Thorson, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.